So you may remember using these glasses in first year chemistry, and they are diffraction grating glasses, and you can see out to the sides it's splitting the light into the different colors. And I'm just shining it at a regular white classroom light, and that's why you can see that there are essentially all the colors of light making up white light because it diffracts into rainbows. This is really probably the simplest and most famous spectra, and that is of hydrogen. And you can see that hydrogen in the visible light really has two lines, the red one and the teal one. The next line starts to get down into the near UV. And this is very cool, but we don't really have any quantitative data here. We just have a red line and a blue line. So let's see if we can leverage our Vernier equipment to get some quantitative data on the wavelengths of these lines so that we can look at the energy involved in the millions and millions of electrons dropping down and releasing these wavelengths of light. So in order to collect data today, we will be using our Vernier UV visible spectrophotometer. And this is a spectrophotometer that is able to read farther into the ultraviolet than most of our other equipment. It reads today by using what's called an optical fiber. This is the optical fiber. Normally when you use a spectrophotometer, you put like a solution in a cuvette and you put it into the slot on the top. But in this case, this optical fiber is going to pick up the light coming into the yellow part and transmit that information through the cable to the UV vis. And that will then go to our Logger Pro software and enable us to make a graph. So now you can see I've turned off the light and turned on the hydrogen lamp, and you may be able to see the shadow of me holding this up close to the lamp to get the data collection. This is the data that I was able to get with the UV vis spectrometer. And you can see I got 3 very clear lines. And I've given you the wavelengths that correspond to these lines. I used the Logger Pro software to isolate the specific wavelength. And let's think about what colors these would be. So in doing so, I have a little image here of the visible spectrum of light, which runs from about 400 nanometers to about 700 nanometers, with obviously individual variation because no two people are precisely the same. So at 661, we can see we're out here in between 650 and 700. So 661, that would be our red line that we saw. And 492, just a little under 500, there's that bright kind of teal blue line that we saw. And in our data, we didn't see the 439 nanometer line. One, it's getting very close to the UV where our eyes are not as reliable. And two, it's obviously not anywhere near as intense as the other lines because the intensity or the brightness of the line is increasing as my peak gets taller. So these were 2 very bright lines that probably drowned out our ability to see this very faint purple line. Just in between 400 and 450, we'd be in the violet range. So I grabbed a screen cap from the video just to remind you of what you were looking at. This pink color was the pink color of the light itself. And then you can see the 2 lines. And out here where the diffraction graded repeated, those 2 lines, the teal and the red line. So we know that thousands of electrons are dropping from a higher energy level down to a lower energy level. and they're releasing energy, which is what we're picking up as this emissions spectra. It's actually possible to do what's called an absorption spectra, and it looks backwards. It looks like the rainbow of light, except at 661, there would be a dark spot where that light was being absorbed. So that's our data for hydrogen emission. And we'll look at the two together in a little bit.
So what I can do this year that I could not do in first year chemistry is get a look at this in a very, very dark room. And this is the emission spectra for helium. You can see it breaks down into several lines. And this is unique for the element helium. In fact, these lines were found during a solar eclipse, and that's how they identified helium as being a component of the sun.
So this is a more complicated spectra because this is from helium. And remembering that we can see from about 400 to about 700, there are a couple of peaks here that we probably could not see. The human eye generally cannot see above 700 nanometers, so we probably did not pick up this 713 nanometer peak. And since we can really see below 400, we probably also did not see the 394. So let's think about the colors that we would be able to classify these. 713 would be at the edge of the visible spectrum into the near infrared or IR light. 673 would be running here right in the dark reds. 593, just a little under 600, would be in the yellows. And then at 507, we'd be seeing something in the blues. And possibly we might be able to see something at 452, but again, those are not anywhere near as bright as the red and the yellow line. So red, yellow, kind of maybe a greenish blue, and a blue. Well, let's look here. Okay, so this does basically correspond with what we saw. Here's the peachy color of the mixed light. And then we break out, we've got a blue, we've got kind of a teal, we've got the orange or the yellow, and we've got a red. So this corresponds to what we saw in our data as well. Blue, green, yellow, and then the red one would be out a little bit beyond the frame of this particular picture. I mentioned that spectroscopy like this is how they actually discovered that there was helium in the sun. And that's because in a solar eclipse, if you remember, there was one of those that we had a partial solar eclipse around here, but there was a solar eclipse that was visible from the US a couple of years ago. And during a complete solar eclipse, scientists used that to study the corona, the outside layer, the bright layer of the sun that you can't see, kind of the sun's atmosphere that you can't see due to the brightness of the body of the sun. And so one of the things that scientists did was they looked at the emission spectra of gases in the sun, because obviously they are extremely hot and absorbing lots and lots of energy. And what you see in that sort of situation, simplified for our equipment, of course, because we don't have something that goes out and covers lots and lots of wavelengths of light. But in this case, this is both spectra overlaid. And so you can see the red spectra for hydrogen is similar. There's the red line and the teal line. You can see they are similar in color, but the wavelengths are different. And in helium, there are peaks where there is nothing at all in the hydrogen spectrum. And so that's one of the ways that they could tell that these were two different elements with two very different electron configurations, is because they saw different energetic transitions. And back when they were discovering lots of elements, they would do things like find emission spectra and sometimes even X-ray spectra and compare it to known elements. If your sample didn't match any of the known elements, you had a lot of proof all of a sudden that you had something that was unique and different. So those are some of the uses for emission spectra. And your job is to pick one of the peaks, the wavelengths in this video, and I've given you the wavelength number, and I would like you to calculate the energy, making sure that you show your work and write it, solve it, math it, of one of those lines, and have that ready to show me in our next class.